5. The last time we covered first subject, cells are fundamental unit of life. Finally, we compare the difference between prokaryote and eukaryote. There are two big differences between them. First one is the presence of nucleus and also presence of the membrane enclosed organelles. Prokaryote cell only has a ribosome, but in eukaryote cell they have the mitochondria and the Daria and Golgi, something like that. Okay, so they are presence of membrane enclosed organelle. Okay, now today we will cover second subject. The first one is the prokaryotic cells are simplest cells. Okay, so this is picture of prokaryotic cell. So this is picture of prokaryotic cell. First one is they are enclosed by cell membrane, like this. This is the prokaryotic cell, and this is prokaryotic cell. They are enclosed by cell membrane. Okay, and also this is the DNA. So DNA is located. The DNA is located in a region called nucleoid. So this is the DNA. So there is no nucleus. So this is the DNA. Very bright region is DNA. So we call it as the nucleoid. Okay. Next, they have the cytoplasm. They have cytoplasm. Cytoplasm is the rest of cell content. Nucleoid. After that, they have cytoplasm like this. And finally, they only have the ribosome. Ribosome looks like the dot, black dot. So ribosome is site of the protein synthesis. It's like this. Okay, so this is the picture of prokaryotic cell, enclosed by cell membrane, and DNA is located in the region called the nucleoid. And they have cytoplasm. Finally, they have ribosome. Ribosome looks like the black dot. And also, in addition to the cell membrane, most prokaryotic cell has the very rigid cell wall. Okay, very rigid. So we have to know about rigid. And finally, we will learn about semi-rigid. Okay, now is rigid. The main difference between the rigid and semi-rigid means that the rigid they are very strong, so the shape cannot be changed. For semi-rigid, they are a little bit flexible. For example, our plant cell wall, they have cell wall is like this. They are semi-rigid, but they are a little bit flexible. Okay, this is the difference. Strong, so it means they're not flexible. Like this. Okay, so they are very strong cell wall outside the cell membrane. Okay. For example, bacteria cells, bacteria cell walls. So let me see the detailed information of cell wall. Okay. So bacteria cell wall contains peptidoglycan layer. So bacteria is divided to two species. First one is gram-negative, second one is gram-positive. So today I will not cover details, but bacteria cell wall, so this is the cell membrane. After that they have the peptidoglycan layers like this. Gram-negative bacteria has one layer of peptidoglycan. However, gram-positive layer has the several layer of the peptidoglycan. After that, some gram-negative bacteria has additional outer membrane is like this. Okay, some have an additional. Some means that gram-negative bacteria have additional outer cell wall. Okay, so it means that this layer is very strong. It's not flexible. Okay. Okay. And also, prokaryote cell has the flagella is like this. Okay, so this is flagella of prokaryotic cells. So if we see the in details, so they have the rotary model, but I will not cover in details, but they are composed of P ring, L ring, C ring. But today I will not cover in details, but the main function is that they used flagellum for the movement. Okay. This is one example. Okay, so if they use their flagellum count clockwise, it means that they can swim like this, count clockwise. But if they use their flagellum in the clockwise, they can tumble it like this. Okay? The flagella contribute to movement, and also flagella can function as addition. So they function as arm, so they can attach to the cell by using the flagellum like this. Okay? Okay. Now we will cover the third subject, eukaryote cell. Okay. So in prokaryote cell we cover flagella and cell wall, and also picture of prokaryotic cells. Okay. Now we will cover the eukaryote cell. Okay. You see here the main difference between the prokaryote and eukaryote is nucleus and its membrane enclosed organelles. Okay. You see here. So this is the animal cell, and this is plant cell. The main difference between the animal cell and plant cell has cell wall, it's like this, but this is the semi-rigid. Okay, they can be a little bit flexible. Okay, that's why for example, this is the plant cell. So they are, when they are very small, is like this, but cell growth is happened, so they can expand. Okay, their vacuole size is increased, so water flows into the cell, resulting in cell expansion. So it means that cell wall is semi-rigid. Okay. Okay, so now we will see the several some example of membrane enclosed organelles. Okay, let me start from the how how to study cell organelles. Okay, we usually use cell fractionation. Okay. Okay. So eukaryotic cell have lots of the organelles. How to study? How to separate cell organelles? Okay. First of all, we put a piece of tissues here, and this is homogenizer. So we use homogenizer to break down cells. So organelles can be separated from one another after cells are broken. After cell is broken, their cell content will be suspended in this aqueous medium. After that, we put this medium in a tube of the centrifuge. After that, the centrifugal process cause particles to sediment, okay, to go down, okay. So during my 14 engineering course, I already teach the calculate the centrifugal force, but today I will not cover in details, okay. But the important thing is that the centrifugal forces in Korean we call it as 원심력, causes particle to sediment. Okay, sit down. Okay. Okay, at that heavier particles sediment at the lower speed. For example, the most heavier particle in our cell is the nucleus. Okay, for example, just only 500 xg. So we applied 10 minutes. So centrifugation at lower speed pellet the nuclei. So after we centrifuge at 500 xg, we can divide it two. One is pellet, the other is supernatant. So in the pellet, most of the component is nuclear. Okay. After that, we can collect this supernatant. Maybe this supernatant contains mitochondria and or ER, Golgi, Ribosome, something like that. After we can only collect this supernatant, and after that we can do the additional centrifugation. Centrifugation of remaining material, this supernatant, at higher speed. For example, 10,000 xg for 20 minutes. We can pellet this is mitochondria. So centrifugation of remaining material at higher speed pellet can mitochondria. Okay, so this pellet we can collect mitochondria. So in this supernatant, maybe there will be ER, Golgi, Ribosome, something like that will be located here. After that, we also can collect this supernatant. After we can apply very higher centrifugation forces. For example, this one is a 10,000 xg for two pellet the ER. We have to apply 100,000 xg, something like that. Okay. Do you understand it? Okay, so let me start from the nucleus. So we can pellet at first time. Okay. 김서진, 이해하고 있어? 안 타고 있는 거 아니야? 안 타고 있는 거 같았는데. 오케이. minus, is it? Minus. Rhea and Marine, is there any other difficulty to hear the class or? Okay. Okay. So I thought that you guys belong to the biology department in your university.
We do, but there is not so much classes in biology in the totally English.
Oh, okay. 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 So last semester you took my epigenetics. So at that time, okay. So this time, this semester you took my biology.
Yeah.
Only ridiculous, but I understand it. Okay. Nucleus. So nucleus is the most heaviest. This is nucleus. Nucleus. So this nucleus. So this is nucleus. Okay, this is nucleus. This is nucleus. Okay. So nucleus is surrounded by nuclear envelope. So nuclear envelope. So this is the double membrane, inner and out nuclear membrane. Okay, nucleus is surrounded by nuclear envelope. Okay. Okay, so and also chromatin distributed within the nucleoplasm. So, okay, we can see the dense region around near the nuclear envelope, and also a little bit the light region around this region. Light region, there is the diffuse chromatin. Okay, so if you see here, this is the nuclear envelope. Okay, so below the inner membrane there is a nuclear lamina. So usually the dense chromatin, we call it as heterochromatin, dark area. And very light region, very light region, there are lots of the euchromatin. So last semester, Leia and Marin took my epigenetics course. At that time, for one semester, we cover what is euchromatin, what is heterochromatin. Is it right? We covered. So heterochromatin is that they are very packed together. So in this location, the gene cannot be transcribed very well, but euchromatin region is not. They are very flexible, so the gene can be actively transcribed. Okay. Take up. Okay. So diffuse chromatin, the right region. So we call it as euchromatin, is located in the nucleoplasm, but I will not cover in details. And also nuclear has a nuclear pore, is like this. They have pore. So nuclear pore controls movement of molecules across the nuclear envelope. Okay, so also in the nucleus, we can see the chromatin fiber, chromosome. So chromatin fiber can be condensed to the chromosome during cell division. So sometimes we can see the nucleosome in the nucleus. Also, nucleus also contains the dense region like this, very dense region. We call it as nucleolus. So in this region, you will see that where the assembly of ribosome begins. So when DNA is transcribed, so they will make mRNA at that ribosome, large subunit of ribosome and small subunit ribosome, they are assembled together. So in the dense, this dark region, so there are lots of the mRNA and the ribosome that are located here. Okay? So previously I covered that here. So in prokaryote cell, you can see the black dot is like this. So this was ribosome. But in eukaryote cell, you will see the ribosome here. So they are black region, dense region. So in Korean we call it as nucleolus. Nucleolus is like this, where assembly of ribosome begins. Okay. So next we will cover also another component of the membrane enclosed organelle. Another example of membrane enclosed organelle. We call it as the endoplasmic reticulum. So this is nucleus. Next to the nucleus, you will find the rough endoplasmic reticulum is embedded with last of the ribosome. Okay. So rough endoplasmic reticulum is embedded with last of the ribosome. And also there is another component, smooth endoplasmic reticulum is like this. So in this smooth endoplasmic reticulum, we call it as SER. So there is no ribosome. So especially the rough RER is a site for protein synthesis because they are embedded with ribosome. But in the case of the smooth endoplasmic reticulum, and also this is a site for lipid synthesis and another chemical modification of a protein happens. So most important part of endoplasmic reticulum is rough endoplasmic reticulum because this is a site for protein synthesis. Next, so this is RER. Okay. Next to the RER, we can find the Golgi complex is like this. Sometimes we call it as a Golgi complex. Also, sometimes we call it as a Golgi apparatus. Okay, anyone is fine. The Golgi complex or Golgi apparatus. The main function of Golgi apparatus. So RER is a site for protein synthesis. Okay, so Golgi complex is receive proteins from the RER. It's like this. After that, so they have this is Golgi complex. Okay, so they have the lumen. Okay, so they can chemically modify the protein in its lumen. Okay, after that, finally, they can target the proteins to the correct destination. Sometimes they can transport protein to the nucleus. Sometimes they can transport to mitochondria. Sometimes they can transport to the chloroplast, something like that. Okay. The main function of the Golgi apparatus or Golgi complex is receive the protein. After that, the chemically modifies protein. Okay. So chemically modification. So the main modification is that. So this is the protein is like this. So they can add glucose and fructose like this, like this. This is the chemically modification. Okay. 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 After that, finally, we will also cover lysosome. Okay, lysosome. So Golgi apparatus is enclosed by membrane. Okay, and also endoplasmic reticulum is also enclosed by the membrane. Is like this. Okay, and also we will also cover the ribosome. So ribosome is the site for hydrolysis. Hydrolysis. What is hydrolysis in Korean? 가수분해. Okay. Okay. So this is a site for hydrolysis of material taken into the cell by phagocytosis. So food particles are taken into the cells by phagocytosis. Okay, so this is the food. It can be taken into the cells by phagocytosis. So there are two kinds of ribosome. First one is primary, second one is secondary. So primary ribosome is originate from the Golgi apparatus. So this is Golgi Complex, Golgi apparatus. So primary ribosome is originated from the Golgi apparatus. So they have a lot of the hydrolysis enzyme. They contain lots of hydrolysis enzyme. At that primary ribosome and second phagosome. Phagosome, they fuse together. So they will make the secondary ribosome. So within the secondary ribosome, the food particle can be digested. So after digestion, some of the undigested material can be released by the exocytosis. Okay. Okay. So after digestion, some of the small molecule can be diffused to the cytosol like this. Okay. So they will make the small molecule means that some amino acid, okay, some carbohydrate can be released in the cytosol like this. Okay. And also we will cover mitochondria. So membrane, there are also membrane enclose their organelles. Okay. The main function of mitochondria is ATP production. So in chapter 9 or chapter 10, we will cover in details. We will cover what is the TCA cycle, what is the oxidative phosphorylation. After that, they will make mitochondria, they will make the ATP. Okay, but today we will cover mitochondria is a site for ATP production. Okay. Okay, they are composed of the double membrane. They are membrane enclose the organelles. So inner membrane and outer membrane. So inner membrane, there looks like this. We call it as crystal. crystal like this. Okay. So usually the crystal contains lots of molecules for generation of ATP synthesis. So this is the inner membrane. So they are they have complex 1, complex 2, complex 3, and the ATP synthase is like this. So this is crystal. So they have this kind of the proteins for protein synthesis. And also they have out membrane. So out membrane is permeable to small molecule and ion. Okay. So some of the after glycolysis pyruvate have to be transported to the mitochondria because the out membrane they are not rigid, but out a little flexible, and they have small pores, so it's permeable to small molecules. But however, glucose is big molecule, so they cannot pass through the out membrane. But small molecule and ion can pass the out membrane. Okay. Now we will cover the chloroplast. So this is a site where light solar energy is converted to the chemical energies. So light energy is converted to chemical energy, means the ATP, photosynthesis of the glucose from the CO2 and H2. So this is the chloroplast. Okay, it looks like that they are composed of the double membrane, inner membrane and out membrane is like this. And they are inside the chloroplast. There are lots of thylakoid. Okay, but today I will not cover in details. But in the chapter 12, chapter 13, we will cover full process of the photosynthesis. Okay, but today we will cover the cell organelles. So eukaryotic cell has the membrane enclosed organelle. The one example is a chloroplast. Okay, also they are surrounded by double membrane is like this. Okay. Now we also cover cytoskeleton. Okay, cytoskeleton is composed of the microfilament and the intermediate filament and the microtubules. So microfilament you will see composed of the actin. Actin plays important role in cell movement. Okay. So this is the nucleus is around here. The cell membrane is around here. So you can see the loss of the microfilament, especially made of the actin. And also, in the cell, we have lots of the intermediate filaments. They are made up of fibrous protein. Okay. So this is a cell, one cell. This is a nucleus is located here. You will see that lots of the intermediate filament is located in the cells. Also, there are another component is a microtubule. They are composed of tubulin. Okay, when cell is divided, tubulin function a very important role in cell division. Okay, so microtubule is the made up of the tubulin is like this. Okay, you can see that this is the nucleus. Okay, so you will see the last of the microtubule near the nucleus, because they are very important role in cell division. Their main function is they are attached to the chromosome and they divide the chromosomes like this. Okay. Okay. The cytoskeleton has 3 functions. Okay. So cell is composed of the last of the cytoskeleton. Composed, for example, here they are composed of actin, actin and myosin. Okay. So the main function of this cytoskeleton, and also cell is composed of intermediate filaments. So it can maintain shell shape is like this, because of actin is located here, so they can maintain the shape is like this. Okay. So, for example, in Korean 공사할 때 철근이 있고 이쪽에 이제 시멘트 하수, 철근 같은 거. Maintain shell shape. So they can mechanical support. Okay, without this mechanical support, they will not shape it like this. Okay. And also this is the, for example, this mitochondria is located here within the cell, but last of the cytoskeleton is bind to the mitochondria. But that's the main function, it holds cell organelle in the position. Okay. For example, in here you will see that mitochondria is located here. So around the mitochondria there are lots of cytoskeleton. They bind to the mitochondria. They can hold their organelle. And also actin and myosin, very important role in the cell movement is like this. So their main function, they have 3 main functions: maintain shell shape, hold cell organelles in the position, and also movement of the cells. And also, now we will cover, we finished eukaryotic cell. Okay? Let me summarize again. So the main function, the main is that they have a membrane enclosed organelle, and also they have the nucleus. Okay? And also they have the endoplasmic reticulum, they are membrane enclosed, and also they have a cytoskeleton composed of microfilament, intermediate filament, and microtubule. Okay, now we will cover 4th subject, extracellular. Extracellular materials provide structure. Okay. For example, especially the plant cell has walls. So this is the plant cell. They have walls. Is like this. So their plant cell wall is like this. Okay. So this is plant cell. Provide rigid yet flexible support for plant. So that's why we call it as semi-rigid. And the cell wall is mainly composed of cellulose. Okay. Okay. because of the cell wall. So, for example, this is the plant cell. Plant cell has cell walls. Okay, that's why. So, for example, some disease organism can attack plant cell, but the plant cell wall can become the barrier to disease organism. Okay, and also because they are semi-rigid. For example, they are rigid, but they are semi-rigid. The rigidity of cell walls limit cell expansion. However, they are semi-rigid. For example, when oxygen is bring to the cell walls, so cell wall becomes loose. That water is flows into cell, so water is deposited in the vacuoles. So after that, it can result in cell expansion because of the plant cell wall is semi-rigid. Okay. However, prokaryotic cells, they are rigid. Okay. So we cover the cellulose. So this is cellulose structure. Cellulose is composed of the beta-D-glucose. So each beta-D-glucose is connected by beta-1,4 glycosidic bond is like this. So beta-D-glucose, 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 like this is cellulose. Okay. Cell wall is composed of cellulose. Okay. And also, we will cover extracellular matrix. Now, our first subject, extracellular material. So extracellular matrix is one component of extracellular material. So this is cell composed of the cell plasma membrane and the cytoplasm. But extracellular cell is surrounded by extracellular matrix. So this is the kidney cell, and also this is the blood cell, blood vessel. Okay, so this is kidney cell. So next to the kidney cell, there are lots of the extracellular matrix. If we magnify this region, we can find the lots of collagen. Collagen is like this, and also proteoglycan. So proteoglycan is composed of protein and carbohydrate. We call it as proteoglycans. So they are mainly composed of collagen and proteoglycan. Okay. So within the cell, so next to outside of the cell, there is the extracellular matrix is located. We call it as ECM, extracellular matrix. Okay. The main function of extracellular matrix, okay, it means that the main function of the cytoskeleton holds the cell organelles in position. For example, mitochondria is the cytoskeleton, so holds the mitochondria in that position. Okay. However, the extracellular matrix So, for example, this is one cell, another cell, another cell, like this. Okay, so outside of cell, there are lots of extracellular matrices like this, ECM, which is composed of collagen and proteoglycan. So their main function is to hold the cell together in tissue. They hold the cell, each cell in position. And also our bone, our bone is mainly composed of extracellular matrix. For example, our bone is mainly composed of collagen and lots of the hydrolysates. So collagen. Okay, so a bone is composed of extracellular matrix, especially the collagen. It contributes the property of bone and cartilage and skin and something like that. And also, this is extracellular matrix. So if we see there are lots of extracellular matrix. They have some holes like this. So it functions as a filter. Filter is like this. So it can filter material passing between the different tissues. So it functions as a filter. Okay? Filter like this, okay? Because they are composed of lots of collagen like this. But they have holes like this. It functions as a filter. So large molecule cannot pass through this filter, okay? Only small molecule can pass like this. Okay. Finally, we'll cover eukaryotic cell evolved in several steps. Now we will cover evolution. So eukaryotic cell has the extracellular matrix. Prokaryotic cell don't have the extracellular matrix, okay? Only eukaryotic cell has the cell wall and also they have the extracellular matrix. Okay? Now finally we will cover eukaryotic cells evolve. They did a lot of evolution. Okay? Prokaryotic cell, for example, if you see the prokaryotic cell, they are very simple. So there is not much evolution, but eukaryotic cell, if you see the complex, they are very complex. It means that there was lots of evolution. So we cover the ER, Endoplasmic Reticulum. So this is the nucleus, and so next to nucleus, the ER is made like this. Okay. So but this is hypothetical. So this is not raw. This is theory. Sorry. Hypothetical evolution of ER. Usually the prokaryotic cell, they don't have the internal membrane, but some cell membrane pulls in the wall. At that further membrane unfolding is happened. So maybe at that this structure become the ER, lobe RER or SER. So this is our hypothetical evolution, hypothesis. Maybe ER came from when membrane pulls inward, maybe this structure becomes the ER. Okay, so this is hypothesis. Okay, so another hypothesis is that a plant cell has chloroplast, The main function of chloroplast is that the photosynthesis. Okay, so but this is the theory of endosymbiosis. Okay, some bacteria, especially the cyanobacteria, especially the cyanobacteria, they can do photosynthesis. So maybe some eukaryotic cells, endocytosis or photosynthetic cyanobacterium. Okay. That the endocytosed cyanobacterium, there retains their ability to photosynthesis. So now it becomes chloroplast. So this is our theory of endosymbiosis. Okay. So after they are digested, okay. So at first time, because the chloroplast, they are prokaryote, they have prokaryotic system, and this is eukaryotic system. They are different. Okay. Maybe at first time there will be no collaboration. But times passed, they collaborate each other. The eukaryotic cell provides some nutrient to chloroplast. And that chloroplast provide, they can do photosynthesis, and they can provide the ATP to the cells. So they can do the endosymbiosis. Symbiosis. In Korean, we call it as the 공생. Okay? They help each other. Okay? Okay? This is a little bit different from the parasite. Endosymbiosis means that they help each other, but parasite, you know the parasite, 기생충, they only accept, they only receive only the nutrient from another partner, but this is endosymbiosis. They provide ATP, but they receive the last of the nutrient from another cell. Okay, but this is a theory. Maybe eukaryotic cell has their structure is very complex. Maybe we thought that endosymbiosis happens. One example is the chloroplast. Okay. Okay, so today we cover the chapter 5, cells working unit of life. So last class we cover the first subject, cell theory, cover the difference between prokaryote and eukaryote. So today we cover prokaryotic cells, and also we cover eukaryotic cell, and we also cover extracellular matrix. And finally, we cover the evolution. Do you have any question?